Okay, so this tutorial is going to walk you through how to do sex-linked crosses and a little bit of information from the worksheet. You should still look over the notes, they're on Edline, and you should still review the worksheet. So the first piece of information tells us that color blindness is a sex-linked recessive trait. The first thing you want to make a note of is if it says it's a sex-linked trait, you always have to indicate the trait as being carried on an X. So it's a sex-linked recessive trait, so that tells me that an X with a big letter, in this case I'm just going to use N, it doesn't matter what letter you use, would code for normal vision, and X with a little letter would code for being colorblind. So now we're going to do our first cross. So you're crossing a colorblind woman and a normal man. So a woman is going to be XX, and the male is going to be XY. And now what's left is to put the letters on the X's and then do the cross. So if she's colorblind and it's a recessive trait, she has to be little n, little n. He only has one X and he's normal, so his X must carry big N. And now if we cross them, I'll put it up here, we're going to see X little n, X big N, Y. And on a sex-linked cross, you want to separate the boys from the girls. Don't just tell me 3/4 have colorblind or whatever. You want to say how many of the females and then how many of the males. So in this case, X big N, X little n, X big N, X little n. All of our females are normal. And then our males, X little n, Y, X little n, Y. All of our males will be colorblind. Okay, and this is a notation that you can make on sex-linked crosses. First of all, it's impossible, if the male is normal, it's going to be impossible for any daughters to be colorblind. That's an important piece of information you should remember. And if a female is colorblind, if she has the recessive trait, she's going to give her X to every son. And since that's the only X he gets, every son would be colorblind. So, our second one, normal parents, they both had colorblind fathers. So, we have our parents again. This time it says they're both normal, so we know that they both have a big N. In the case of the male, he only has one X, so whether his father was colorblind or not makes absolutely no difference, because his father would have given him the Y chromosome, not the X. But it is important to know that the mom's father was colorblind, because that tells me about her other X. She's normal vision, she must have gotten an X with a big N from her mom, but since her dad was colorblind, he would have given her X little n. And now we just do the same exact cross again. X big N, X little n, X big N, Y. And we look at our outcome. X big N, X big N, X big N, X little n, X big N, Y, and X little n, Y. So in this case, all of our females are going to be normal. But this time, half our males will be normal and half of our males will be colorblind. All right, in our last cross, we're given the offspring and a little information about her mother, and they want to know the genotypes of both of her parents. So first step, if she's colorblind, we know her genotype. She's X little n, X little n. If her mother's normal, her mom definitely has at least one big N. If we work backwards, we know she had to get one of her X's from mom and one of her X's from dad. That tells me her mom's other X had to be a little n. It also tells me that her dad had to be colorblind, because that's the only way she would have gotten another X little n. So that's sort of, I guess, 3 examples of sex-linked cross problems. They get progressively harder, but you'll notice that you're doing the same thing over and over again. It's just really how you interpret the results based on the trait. All right, this is from the worksheet. It's just a little explanation of haploid versus diploid. So you need to be able to tell me, in this case, or for these, which one of these 2 choices in each of these statements is the choice that's haploid. Before we go through those, just a reminder, think of di as meaning 2, so that means that diploid refers to having 2 copies of each chromosome. haploid refers to having only one copy of each chromosome. Why would a cell only have one copy? Well, there's only one reason why cells end up with just one copy, and that is because of the process of meiosis. In meiosis, you might have a cell that starts off with 4 chromosomes. They line up side by side, and what's going to happen is each cell is going to get one copy of each chromosome. And this is how we make gametes. In fact, it's the only cells in a human that would be haploid would be gametes, or sex cells. So now that you know that, let's go back over here to this. So somatic cell versus sperm, it would be sperm. Sperm's a gamete. A somatic cell is a body cell, which is going to have 2 copies of each chromosome. A cell after mitosis or a cell after meiosis, I just explained to you that the process that makes cells haploid is meiosis. Mitosis makes cells that are identical to the original cell. It's what happens in body cells. 23 chromosomes are 23 pairs. Again, 23 chromosomes. When they're in pairs, we're talking about a diploid cell. And finally, a cell with XY, or a cell with either X or Y, it would be the cell with either X or Y. If you think of a male, he has XY. But when he goes to make sperm in meiosis, his sperm is either going to get an X or it's going to get a Y. All right. Next thing, I told you guys that you needed to know the difference between the position of the centromere on the chromosome, which is basically what this is talking about. The easiest one is metacentric. Meta means middle. So if the centromere is in the middle, that's going to be C. Submetacentric, it's somewhat off-center, if you think of it that way. Notice the top is shorter than the bottom. And then finally, acrocentric, which the worksheet I believe called parasubmetacentric, is basically where there's almost nothing. There might be a little something on the top, but pretty much the centromere is at the end. The next one, this is just parts of a chromosome. So telomeres, for starters, think of like telophase being the end of mitosis. Telomeres are the ends of the chromosomes. Centromere, this one, there's only one. And the centromere, not necessarily in the middle, but it's what connects basically the top and the bottom. Finally, P and Q. The worksheet explains that the shorter arm is P and the longer arm is Q. The truth of the matter is, whenever you see a picture of a chromosome, if they're putting it right side up, they always have P on top. So even if it's a metacentric chromosome with the centromere right in the middle, it doesn't matter. The top is always going to be your P arm and the bottom is going to be Q. How do we know which bands code for the most stuff? Well, when they make a chromosome karyotype, they dye the chromosomes. The dye sticks very, very well to areas of the chromosome that are really coiled up. And if they're really coiled up, that basically means they're inactive. It means that they're coiled up like a closed book, which means they can't be read. And chromosomes have to be read. The genes are read in order to make proteins. So the part of the chromosome that would be coding for the most stuff would be all of these parts that are white. If it's gray, it's somewhat coiled up. If it's black, it's really coiled up. And that would mean that those genes, at least at the time that the picture was taken, are inactive. All right, last question. This is one more sex-linked problem, but this time we're talking about a trait that's dominant. Your cross is going to pretty much look the same, but how you look at the phenotypes of the offspring is going to be different. If it's sex-linked dominant, we're going to use we'll use X big D to represent the disease, and X little d to represent not having the disease. So here we have a woman that has the disease. That means she's definitely got a big D. Since her father was normal, he didn't have the disease, he had to be X little dy, so her other chromosome must have X little d. She marries a man who is normal, so he's going to be X little dy. And now we do the cross just like we did before, X big D, X little d, X little dy. And what you're going to get this time is X big D, X little d, she would have it. X little d, she wouldn't. X big dy, he would have it. And X little dy, he wouldn't. And so basically you end up with a 50% chance of having it in females and also 50% in the males. It turns out if you mess this up, you actually get the same answer, because if the trait was recessive, you would basically see the same thing. It's just that the one that would have it would be this one versus this one. I also added in class one more question about this where I flip-flopped it, and I said, well, what if the same woman, X big D, X little d, married a man who had it? And that would look like this. So we'd make our Punnett square, X big D, X little d, X big D, Y. And this one, if you mess up your reading of the boxes, you will get the wrong answer. So here's our girls, and in this case, they both have it. If the trait was recessive, actually, they would both be normal. And then for our males, X big dy, X little dy, so 50% of the males would have it. So it really doesn't change our answer for the males, because remember, it only mattered what the mom had for the males, so changing the dad doesn't affect them, but it does matter for the females. Okay, so hopefully that was helpful. I highly recommend you maybe use the Mastering Biology as practice, and also you should go to the notes that I posted and look at the tortoiseshell cat crosses, because in that case the trait is codominant, and it does affect what your outcome looks like if the trait is codominant versus dominant or recessive, so review that.